So our next session for today is PPMD moving the needle. And so this is an opportunity for us to share some of the research that PPMD is funding. And we're able to do that because of, you all in the room, your contributions, your grassroots efforts. So this is really how you are helping us to move the field forward. And so the first two talks you're going to hear are from companies that PPMD has invested in through our venture philanthropy program, where we provide some seed funding to early stage companies to try and help them grow and accelerate and bring therapies to the clinic. So our first speaker for this session is going to be Dr. Casey Childers with Kinea Bio, who's talking about a next generation gene therapy that they're developing.
Hey, thanks very much. It's such a great honor, and I'm humbled to be here to speak to you. Kinea Bio is the name of our company. I'm a co-founder, and I serve as the chief executive officer. We're focused only on neuromuscular disease. That's all we do, and that's all that we've worked on for 30 years collectively. We are a startup, means we have not much money. But we started in 2020, founded out of the University of Washington in Seattle. And our timeline really goes back way before that. Our principal scientific founder is Jeff Chamberlain, Professor Chamberlain. He really was the first to invent microdystrophins for D&D gene therapy back when he was at the University of Michigan with Francis Collins. So in the 1990s is when this started, and I should say that I think it was either 95 or 96, Pat Furlong provided a fellowship for me to step away from the clinic for a period of time and work on a research project on Duchenne muscular dystrophy. And that really kick-started my career. Now, almost 30 years later, we are very fortunate to receive some pre-Series A funding from PPMD to initiate an IND-enabling work for our lead program in Duchenne muscular dystrophy. I'll talk about that very quickly. So our platform technology is focused really on AAV, gene therapy, adeno-associated viral therapy. There are sort of two areas. One is targeting, the other is splicing, and that's shown in this cartoon. So in terms of targeting, meaning we want to steer genetic cargo to where it needs to go accurately, precisely into the skeletal muscle, into the heart, we have a collection of muscle-specific promoters that were developed by Professor Steve Hauschka. He has done nothing in his entire career but 50 years working on steering a genetic cargo to skeletal muscle and heart using these proprietary promoters. The second part is that we have in-licensed some second-generation engineered capsids, meaning these are AAV capsids that have been either selected or engineered to provide more specific, accurate dosing to the skeletal muscle and heart, and also at the same time decreasing the stress on the liver, which means that we can more safely and effectively deliver genetic cargo. And then the third component is what we're really, really excited about, and that's a proprietary technology that we've called Simply GT, and it enables the delivery of a gene product that exceeds the current AAV packaging capacity. I'll tell you what that looks like. This is our lead program. PPMD has invested in this. We call it KNA 123. And you may know this, but human gene therapies using AAV vectors have demonstrated incredible promise. One already received accelerated approval in the clinic. The challenge is that with these viral vectors, there's a loading capacity of about 4.7 kb. And that excludes genetic disorders with other larger genes. And there are many like that in the disease spectrum. The Duchenne muscular dystrophy coding sequence is more than twice that. It's 11.2 kb. So how do we overcome that? Well, people like Jeff and others have engineered these, what we call microdystrophins. These are microdystrophins that can replace the functional effect of dystrophin. And as you know, the progression of DMD goes from muscle damage, degeneration, loss of muscle tissue, and a replacement by fibrosis. But what we have come up with, and it was discovered many, many years ago, is to use something called inteins. So inteins are found in nature. They're found in microorganisms like virus and bacteria and yeast. What it enables engineers, scientists to put sticky ends on two different genetic cargo and then use two separate AAV vectors or other type of vectors to bring them together in the body where they're transcribed and then translated. And with these sticky ends, the inteins come together, they bring the genetic cargo and fuse into a large protein that's what's ligated together. Then the intine removes itself from the cargo. So this is what we've been working on for about 5 years now. And that's a dual vector strategy to deliver something we call a MIDI dystrophin. This is not a microdystrophin. This is essentially two microdystrophins put together, and it forms a much larger product using this intine technology. Well, how big is it? It's not the entire folding dystrophin, although you can do that. It's twice the size of a mini dystrophin, but it's the same size as the dystrophins found in patients with specific mutations where they have no clinical symptoms. No clinical symptoms. What's our proof of concept look like? We have done extensive work in mice so far. Paper will be published in the journal Nature in the next 3 to 4 weeks, but these are the highlights here. When we use this MIDI dystrophin split intine technology and deliver it with an AAV vector, we've compared that with a microdystrophin using either an engineered capsid or an AAV9 capsid, or the MIDI dystrophin together. And the graphics here, I won't go into detail, but bigger is better. Better force, better effect on heart function. So in our proof of concept study, we find highly functional MIDI dystrophins are produced using this system we call simply GT. The effects are better than when you use MIDI dystrophin with more of a natural serotype like AAV9, but if we use this myotropic, muscle-specific vector, it's better. We found no signs of toxicity when individual components were administered, but by far, when we compared to AAV9 with microdystrophin, the MIDI dystrophin outperforms it by at least a 10-fold lower dose. So we can reduce the dose by 10-fold, achieve superior effects, and we think this is going to be a significant improvement in the clinic. So this is if you would, a third-generation gene therapy. So this is my name. I will talk to anyone. Please send me an email, call me. I'm happy to engage. And again, I'm honored and humbled to be here. So thank you so much for your time.
Thank you, Casey. Our next speaker is going to be Frank Gleeson from Satellos Biosciences. So this is one of our earlier investments, and they have a novel approach for trying to treat Duchenne. So I'm going to bring Frank up.
Thank you, Eric. And good afternoon, everybody. We're really quite pleased to be here and very grateful for the support that we received from PPMD, not only because it helped us financially and helped us at a time, particularly during COVID, when it was difficult to attract financing, but also because of the inspiration that it provides to us. Every time I attend one of these events, I'm just overwhelmed by the strength of character, the determination, and the courage by all of the family members that I have the pleasure to meet with. And we want to help. We want to help because we've made a discovery related to Duchenne that is different, new and different than you will hear from any of the other scientists and companies that are talking about this disease. We believe that we're working on the next horizon of this disease, which is the idea of regenerating muscle, bringing muscle back. And I'll tell you how we plan to do that and where we are in development. So we're a publicly traded company, and therefore you must read this and memorize it so that you can, be on side with the regulators at the SEC. What we discovered is that Duchenne is also a stem cell disease. It is not only a genetic condition, it is a stem cell disease as well. That's a consequence of the genetic situation, and it doesn't make things more scary. It actually is, we believe, good news, because it means there's another avenue to be helpful and to treat this disease. And what we're doing about it is we've invented a drug, a small molecule in tablet or liquid form that can be ingested by mouth that we believe has the potential to fix this stem cell problem and bring muscle back. My colleague and co-founder, Dr. Michael Rudnicki, published a landmark paper in Nature Medicine in 2015 describing this situation, and we now, over the last several years, are seeing ample evidence that the condition that he identified in the MDX mouse also exists in human. So this really encourages us that our approach could make a big difference. So this is a really simple idea, a pill to regenerate muscle. What you're looking at on your left-hand side is the actual pill that we have developed. That is our pill. We call it 3247. It has taken us 6 years since we formed the company to get to this point and having that pill. And there are two things I'd like you to be really cognizant of in what we're trying to do. Number one, this is independent of dystrophin. We are not trying to put anything into your son's or your daughter's bodies. There is no infusion, there's no toxic side effects because of any immune reactions. It's a pill, like a Tylenol. Whereas the gene approaches, the genetic approaches, are focused on dystrophin and putting it back in the fiber to stabilize the fiber. We are focused on regenerating the fiber, restoring muscle. These can be massively complementary. So our approach can be standalone if your child has an AAV antibody challenge, or add-on. So this is something that we believe has the potential to be, frankly, revolutionary. So why we're so encouraged by this is that the problem we identified is that in the bodies of individuals with Duchenne, they are unable to make new muscle cells efficiently. And what you're looking at on the left-hand side in those graphs is proof that in human beings, their biopsy material, at a wide range of ages, including ages when they're no longer ambulatory, these children continue to have stem cells in green. What they lack in purple are the new muscle cells. Their stem cells are not creating new muscle cells efficiently. This is what we discovered in mouse, and this is now true, clearly true in human. And so now that we know that, we designed a drug to fix that problem. So I'll give you some biology very, very quickly. I know time is precious here. On the left-hand side, you're looking at a muscle fiber. Those little peanut-like shapes, those are the stem cells. Those are muscle stem cells that punctuate through the fiber and live in kind of niche environments. When damage occurs, and damage is always occurring to all of us, quite frankly, through normal wear and tear, let alone any extra damage and extra pressure, our stem cells leap into action, literally leap into action, and that's what that blow up is showing. And they do it in a geospatial way by orienting themselves to the muscle fiber at a 90-degree angle. So if you're not a geometry person, or that's just up and down, north-south. And when they're in that space, Dystrophin in that stem cell tells it to create two cell types, to create a muscle progenitor cell. When dystrophin isn't there, which is on the right-hand side, our bodies would make more stem cells, but not new progenitor cells. And so that's the problem we're trying to fix. And fundamentally, think of SAT3247 as replacing that dystrophin signal. Other people are working on the fiber. We're working on the creation of new fiber. It's that simple. It's a lot of science. My colleague, Dr. Rudnicki, is a world authority in this area of muscle stem cells. He discovered them. He's been working on this for 30 years. He started at MIT with Dr. Rudy Yanish, who's a famous biologist. He has over 250 publications in this field, is now a member of the Royal Society of the United Kingdom, and if you look up his webpage on the Royal Society, they'll basically call him the father of muscle stem cells. So this is deep, deep science. I'm almost done. I want to show you, in mouse, we correct the stem cell problem. On the left, we correct the stem cell problem. On the right, it increases the force generation capacity of the animals. And in dog, this is new data that just literally came to us in the last couple of weeks. We're working with Dr. Dongsheng Duan at the University of Missouri. This just blew our minds. You're looking at biopsy material of one limb of an animal, a dog, before being treated with SAT3247. What you're looking at on the left is the damage area. That's dying tissue as stained by immunoglobulin, which is a signature antibody of the immune system. 4 months of treatment, we've restored that limb. We have more strength in those animals. So we now know what we've seen in the mouse translates to the dog, and we're highly excited about its possibility of translating. And I'll wrap up with this slide just to let you know we are about to begin clinical trials, a phase 1 safety trial in healthy volunteers this year, and early next year we will begin in patients, in Duchenne patients. Our plan as we move along is to do separate studies both in ambulatory children and in adolescents and young adults that are non-ambulatory. So please pay attention to our website. We will work closely to keep PPMD informed, and we hope very soon that we'll be able to help your children. So thank you very much, and thank you, PPMD.
Thanks, Frank. I think it's also important to recognize that PPMD funds research that isn't just there to look at new potential therapies. We also have questions that we need to help understand and answer, and so that's, I'm going to have Dr. Stan Nelson from UCLA come up and talk a little bit about his research that's helping us to better understand the disease and what happens when we're treating with some of these dystrophin restoration therapies.
Very good. Thank you. Thank you, Eric, for inviting us, and thank you all for supporting this research. This research, I want to point out, is an equal collaboration between myself and Dr. Michelli at UCLA, and has been funded critically by some of the pilot funding from Parent Project. So our goal at the end of the day is to try to much better map out the atlas of the complexity of what Duchenne muscular dystrophy is. So you've already seen from a couple of the other presentations, it's not just the myofibers, it's all the other cells that are present in muscle, and we need to know more about them. So this is a project to do that in a fairly grandiose way that really requires partnership with many of you in the room, and many of you have already partnered with us. So our goal at the end of the day is to identify new biomarkers and new therapeutic insights for how to augment things like dystrophin replacement or improving satellite cell function. So we do this through biopsies. So we must be able to get at the muscle, and we must be able to sample the muscle This slide is just simply reminding me to show you the gray and whitish samples are skeletal muscles, 3 different samples, showing that muscles look different, right? Sometimes have more muscle, sometimes there's more fat, sometimes there's more fibrosis, but they all have characteristic features of a dystrophinopathy, of a Becker muscular dystrophy or Duchenne muscular dystrophy. And through imaging DMD, we can look at many, many images, and they've been able to determine which muscles are affected at which rate. So we can now select muscles that have a larger amount of muscle fiber preservation or more damage. And we can do that intelligently as we look and biopsy boys who are older and may have less muscle remaining, and successfully biopsy muscles from boys that are younger as well. So we've pushed forward with a core needle biopsy technique that gets us sufficient muscle sample that we can look at typical clinical histology, see those very, very well, quantify dystrophin, but also make cultured cells in the lab so we can culture out lymphocytes, macrophages, fibroblasts, muscle stem cells, and have all those available for further study and study the tissue, and now explore at single cell resolution all of the cells that are actually present and what their expression changes are being in muscle. So on the upper right is the result after one of the core needle biopsies, which is about a 2-millimeter scar through the skin to sample in about a 30-second actual procedure for the actual sampling, but it takes about 10, 15 minutes to numb up the area relative to what we do with an open biopsy on the other side. We've done this now on actually hundreds of individuals. We've analyzed data from dozens of individuals, and this is a subset of the data. So in the colorful graphs in the middle, on the left is hundreds of thousands of nuclei collected from dozens of individuals, where we can use the nuclei themselves and their gene expression to recreate what did the muscle look like. So each nucleus from a muscle fiber expresses different genes than a fibrotic cell or an immune cell. So we can use that information to say, and group those cells together and find all of the major cell types of muscle. And blue is the myofibers. And then we can look on the right, did those muscle samples come from somebody from Duchenne or from healthy? And we can see substantial difference in gene expression in the myofibers, and that a little bit of dystrophin in those muscle fibers push them back towards a healthy state. So we can now use this information to say what is changing in the muscles, and we can do that across every cell type. We can also, everybody who comes to participate in this study will ask you for North Star information, ambulatory information, and we can indeed see when we can see some low levels of dystrophin, there's often a more mild disease phenotype, still in the Duchenne spectrum, but more mild. So I don't think I can point very easily, but in this graph, this is showing things, some that we already know. For instance, in the far left are myofibers, and myofibers are decreasing in Duchenne. So when you have Duchenne, you have fewer myofibers, and we can see that in this data. The red is Duchenne, blue is healthy, and it's decreasing. And instead of myofibers, the muscle that our boys have is expanded in other cells that are causing problems. One is fibrocytes or fibroblasts, which are greatly expanded about 3-fold in Duchenne muscular dystrophy, and immune cells, an example is myeloid here, which are also increased substantially in Duchenne, contributing to an inflammatory state and fibrosis within the muscle that's important to be treating, and that's a very complex process. So what we're trying to identify is all of the biomarkers that you could find within muscle and all of the way these cells are talking to each other. So in this particular instance, in the upper right is healthy muscle, and there's a 5 there. That's the type of fibroblasts that exist in our healthy muscles. And when we have dystrophic muscles, those fibroblasts aren't just the same fibroblasts, and they didn't just grow a little bit bigger. They're wildly different fibroblasts with different characters, and they've changed, they've diverged into multiple different cell types. So one is fibroblast 6, we call it here, and it's an inflammatory fibroblast that has new characteristics that are driving inflammation in the muscle and that are communicating with immune cells, communicating with muscle cells, communicating with satellite cells to promote this dystrophic environment, and they provide many, many targets for therapeutic intervention. So an example of what we can learn from this is across that fibroblast 6 population, because we've converted muscle into data, we can now search that data across all data in the world, and we can find those fibroblast 6 population are very, very similar to a fibroblast set that's in the joints of inflamed arthritis joints. So now we can match data from arthritis to Duchenne muscular dystrophy and say, are our fibroblasts similar? Could we use therapeutic strategies that are similar, and are they driven by similar biological processes? And the answers to those questions are yes, we can. And we can even make cell models from this. So this is a simplified version of things for what it looks like. So on this graph are fibroblasts, some of the mononuclear immune cells, and the myofiber and the satellite cell, and they're all talking to each other. They're sometimes hearing good messages, sometimes hearing bad messages, and we want to change that to actually dampen all the bad messages. So this is getting to the issue that we've all been talking about for a while of combination therapies, and then how will we use those? This type of data intelligently lets us know how we're going to be amalgamating some of these therapies. We're starting to look at post-gene therapy patients, and we can see the transgene being expressed. We can see it at the nucleic acid level, and we can see durability of expression out several years. We can see changes in these fibroblast populations that are toxic to the muscle in some, and we see a lack of that change in others. And we can also expand out the immune cells and then ask, are immune cells playing a role? And the answer is, in some people post-gene therapy, absolutely, and in others, not. And so we've been funded by PPMD. This is actually now leveraged into a Department of Defense grant. Actually, the other Department of Defense grant was given to Kevin Chamberlain, I could see on their slide. So your funding is going to good purpose, and we have now funding to collect about another 32 samples for those participants who've been post-gene therapy, and we'll put it to good work in this. And we hope to learn a lot of new information about therapeutic targets and biomarkers in that process. And we thank you very much.
Thank you, Stan. Another area that we, I think hearing a lot of the gene therapy talks this week, you've probably heard about neutralizing antibodies, and this is an area that we obviously want to try to address, and I think this was a really great project because ourselves, MDA, and Cure Duchenne were able to all come together and provide co-funding to Dr. Byrne from the University of Florida to look at a strategy to try and address neutralizing antibodies so we can expand who can receive that therapy.
Thanks very much, Eric. And I know we're going to move quickly through this, and I really do appreciate the level of cooperation and consideration from all the organizations, and especially now that we're well into this project, the cooperation of argenx, who's the manufacturer of one of the products that we're going to study to modify antibodies. Just to give you an idea of the importance of this problem, at least in this small study done by Sarepta, about 17% of patients were seropositive or excluded from treatment with the Elevidys gene therapy. In our experience, about 400 samples we've screened and kids who were trying to enter trials, it's actually more like a third. So 1 out of 3 of you has a child who might have an antibody level that's exclusionary, where it may be amenable to a treatment very near the threshold that can be lowered. We compared the nature of AAV9 antibodies, which have been tested in about 3,000 individuals with SMA, to those with for RH74 by using the same assay, and they're really nearly identical. Importantly, the antibody levels increase in school age and in young adults. What happens to an AAV vector that's delivered in the presence of an antibody? What's important to understand is that the antibodies can completely block the therapeutic effect of the vector by retargeting it away from its intended target in the muscle or in the heart or diaphragm. And because of that, we want the lowest level of antibody at baseline possible, and importantly, to probably decouple the peak of the antibodies that are formed with the delivery of the vector, because even after delivery, the nature of the antibody response influences the ultimate outcome. Just so that you all have resources to understand how these tests are done and how they're interpreted, we published with colleagues at Pfizer a natural plain language summary of the concept of antibodies and what they mean for gene therapy and how the tests are done. So you can see the reference there was published last year. And in a more detailed paper related to that plain language summary, we characterized the findings about antibody assessments done all over the world. And you can see that there are some stark geographic differences in levels of AAV antibodies that either may relate to immune response genes or they may relate to environmental exposure of AAV vectors. Importantly, I think I want to emphasize that even though we talk about the different types of AAV, whether they're myotropic AAVs or the ones in use today, they're all based on a very common background of parvoviruses that make them nearly identical from the standpoint of the immune system. So antibodies against one vector are often found commonly against others. Two tests are used and commonly, and you've heard about neutralizing antibodies. That test is done within a type of indicator cell that looks at the antibody's attachment to the cell and transfer of a marker gene to that cell. That test was predominantly being used in the Pfizer studies, and another approach, and that's called a transduction inhibition test. The other test that measures total antibodies is more commonly used and is probably a little more discriminating at the lower limit of detection, which is really where these thresholds exist. Because individuals who might have a titer of 1 to 100 and allowed to receive gene therapy, after gene therapy, their antibody titer is in the range of 5 to 10 million. So that's the level of increase. And we ultimately also want to use what we learned from this study, supported by these three organizations, is how not only pre-treatment antibodies might be influenced, but how we might use those in combination with other techniques to allow for re-exposure of AAV vectors to maintain the effect lifelong. There are lots of places where those interventions can take place. I'm not going to go through all these in detail, but in addition to this VYVGARD study that I'm describing, we're actively working on ways to develop central tolerance influencing the overall lifelong responsiveness to an AAV vector. We're taking advantage a little bit of riding on the coattails of another disease problem called myasthenia gravis, which is mediated by antibodies. And companies like argenx and UCB, a European drug manufacturer, have developed ways to lower antibodies that impact the both complement activation and in-body influence on the place where muscle transduction happens at the neuromuscular junction, and this causes weakness in myasthenia. So the use of a drug for myasthenia in Duchenne is a kind of drug repurposing that we talk about a lot. And the drug we're using is called VYVGARD, or efgartigimod alpha. It has been used predominantly in adults, so one of the challenges is to find ways to identify the correct dose for children. VYVGARD blocks antibody recycling, which is a way that we maintain the levels of antibodies in our blood. So when you add VYVGARD into the circulation, and it's usually given in weekly infusions, 4 of them, the amount of recycling is reduced, and therefore less of the antibody enters the circulation. And we know that it works kind of like this. You can see that the VYVGARD in that crystalline structure gets inside the cell along with the antibodies to the areas in the lysosome where antibodies are recycled and in some cases degraded. If you prevent them from being recycled, they go into the degradation pathway, and then the levels of antibodies are reduced. And in some cases, we think that even after 2 infusions of this drug, the amount of antibody can be reduced by 80%. Here's an example of that with another product that we're going to add into this study called Rustigo. This is made by UCB. We studied this preclinically in animals and showed that we could not only block and lower antibodies that were from pre-administered to the animals, but that it persisted after the vector was delivered. Here's an example of how this approach was combined with other antibody-lowering strategies to even further reduce the antibody levels. This is the study design that's intended with one group that have naturally or environmentally acquired antibodies and a second group that received previously gene therapy, and will do safety follow-ups for a period of time after, tracking the durability of the lowering effect. And then this will start with a group that's been pre-identified to have elevated antibodies, hopefully ultimately be able to expand this study for clinical use now that there is commercial approval of a gene therapy product. And I'm going to end by going over how we might be specifically able to lower just the AAV antibodies in a study being done in collaboration with Biohaven. We're taking advantage of a molecule that can bind to the antibody, drag it into a degradation pathway in the liver, and specifically lower the antibody level. So this is called the MODE platform, or molecular degraders in the extracellular proteins, and we're working with Biohaven to develop a second-generation product that will bind specifically to the AAV antibodies and not reduce all antibodies. So this works actually by a very simple subcutaneous injection given twice before the gene therapy. And here's an example of the work that Biohaven's done thus far to generally lower antibodies, and we hope that we can find the specific antibodies that most impact AAV transduction. And then I'll point you to this comprehensive paper that we published in January, which describes all of the immunologic events post-exposure to AAV. And what I'm trying to emphasize here is that if we can unlink the peak of the AAV in the blood with that of the onset of the antibodies through one of these mechanisms, this actually increases the efficacy of the gene therapy, and we see up to 2 to 4 times more expression in the absence of antibodies than when given with just glucocorticoids alone. So that's what my update for you, and I'm going to thank again PPMD, Cure Duchenne, and the MDA for supporting this work, as well as the Duchenne UK Foundation and Kindness Over Muscular Dystrophy. So thanks very much, Eric.
Thank you, Barry. And again, we're really thrilled to be partnered with those other organizations to try and address what are, these really prevalent challenges in Duchenne. And our last speaker for this session is going to be Dr. Rita Perlingeiro from the University of Minnesota. And again, this is a collaborative effort that we are funding with our friends Duchenne UK as we look at another novel approach to try and treat Duchenne. So I'm going to bring Rita up.
Good afternoon, everyone. I would like to start by thanking PPMD for the invitation to share our progress and plans with you today, and for the support over the years, in particular now as we embark in a phase 1 clinical trial. And I would like to thank all of you, patients and family, because you inspire us every single day to do better and to do faster, as fast as you can. So I'll try in the next 5, 6, 7 minutes to tell you where we are in our journey to begin a phase 1 clinical trial to test Myopaxon, which is allogeneic cell product derived from induced pluripotent stem cells to treat Duchenne muscular dystrophy. So to begin, why muscle replacement? So this is a therapy focused on cell replacement. So cell replacement because in the end of the day, even if you are able to induce muscle regeneration in a genetic disease like Duchenne, the final product, the muscle fibers that will be formed from this regeneration, it still will be mutant. It still will be lacking dystrophin. So in any strategy that you promote cell replacement, you are providing healthy muscle and hopefully muscle stem cells to the diseased muscle. So that's the goal here. Next. This one. Okay, so I have a very simple disclosure, so not very long as you can see. I started a company called Myogenica that hopefully is going to take Myopaxon to the next phases of treatment. So what's myopexon? A little bit of cell biology here for you. So myopexon is myogenic progenitors derived from pluripotent stem cells. You might have heard about these cells before. So pluripotent stem cells, which include induced pluripotent stem cells, which are these cells that you can reprogram from any cell type in your body, are cells that can be expanded indefinitely in the Petri dish, in a culture, in the lab. What's the very nice feature of these cells, not just because they can be expanded indefinitely, is that actually they have the potential to make any cell type. So this is great, they can make any cell type. So we want a particular cell type, right? We want skeletal muscle progenitors, meaning cells that can make muscle fibers and muscle stem cells. So many years ago in my lab in the University of Minnesota, we tried to understand if you could, these cells that can go any direction, can you instruct them to make only skeletal muscle? And can you make lots of them? So we were able to, we have, if you look at our website or in Myogenic, you will see a list of publications. I'm not gonna go on all those details because I'm gonna take you to the path that took us here to a phase 1 clinical trial that before I forget, we received FDA approval exactly 1 month ago. So we are very excited and this very recent news, and I'm happy to share with you. So this strategy we have developed many years ago allow for this generation, as you can see in the scheme on the right, very large numbers of these cells that could make anything. Now they can make only myogenic progenitors, and you can make lots of them, which is very important for therapeutic application. But importantly, when you transplant these cells in animal models of many different animal models of Duchenne muscular dystrophy and other types of muscular dystrophy, they make functional muscle, and they also give rise to muscle stem cells, allowing for long-term regeneration, so indicating they are able to contribute to multiple rounds of regeneration, which is very important for long-term effect. Okay. So after then a lot of proof of concept in the lab, more than 50 publications in this area, 8 years ago, we decided, okay, we know a lot about these cells. What would it take us? Would it be feasible to take this to the clinic? So is it doable, right? So we collaborate with a number of investigators in the University of Minnesota and outside, and we have translated the whole protocol to be compatible to clinical application, which took several years. The process was totally adapted to be done in a clinical grade facility and not generate a few millions as we need in the lab to inject mice, but actually billions. So the cartoon you can see, we started, as I mentioned, an allogeneic cell product. So these cells can be injected in a number of patients. These cells are derived from an iPS cells from an unaffected donor. In this case, it was cord blood. And we generated a source cell bank. So these are iPS cells that are expanded in small quantities at this early stage in this clinical grade facility. We modified these cells with our technology to be able to instruct these cells to make only muscle and lots of them. And in a very abbreviated scheme, you can see we have vials of the master cell bank, which is this modified induced pluripotent stem cells. We have more than 100 vials in the freezer. And as you all start the differentiation, you have a working cell bank you can freeze. It's an intermediate. We have more than 50 vials. And one vial of this working cell bank gives rise to the final product, more than 150 vials in this case. In each vial contains 50 million cells. So you can do the math. We have billions of these cells, clinical grade in the freezer, ready to be used for multiple clinical trials if you have the success in the phase 1 trial. Very importantly, that all the safety studies were done with the same cells that are in the freezer that are ready to be used in the phase 1 trial and subsequent trials. So all the way through the process, we have quality control. All the tests have been scrutinized for safety. And we have tested the efficacy in different models of muscular dystrophy, including the MDX. And we have tested safety in lots of studies in mice and non-human primates. So mice allows us to do very long-term studies over a year. and looking biodistribution, toxicology to assure the safety of the cells. But very nice, we could move into non-human primates because, as you can imagine, it's much more similar to us, and you could actually inject very large numbers of cells intramuscularly and look for toxicity in cell numbers very similar to what actually will be used in the phase 1 trial. So all these studies show the cells are safe. And this puts us in the plan, and these studies will be led by my colleague, Dr. Peter Kang from the University of Minnesota. He's the director of the Greg Marzoff Jr. Muscular Dystrophy Center. And in this phase 1 trial, as you can imagine, the goal is safety, right? So for safety, they are injecting the EDB muscle, it's a foot muscle, and we're going to be a dose-escalating dose, as you can see here, and the primary goal is safety, so you can move fast to important muscles. So our goals here, as I mentioned, is to determine the maximum tolerable dose of intramuscular injections of a single dose, determine, the safety in animal models. You want to see the safety in humans, measure engraftment. Do you see dystrophy? And after the transplantation of these healthy cells into the muscle, measure any immune response and determine feasibility and tolerability. So who can enroll in this phase 1? Again, because it's safety, what FDA allows us to do is to go into patients with Duchenne that are age 18 years and older, no ambulatory, and this is the EDB muscle of the foot that's going to be injected. These are the steps of the process. I know you already stood up, so I'm going to speed up. And what I want to end in this important slide for future, even though you're still in the present, is that you want to, it's like I tried to say, you want to do better and faster, right? But we have to follow safety instructions. And you want to make sure that before you increase the number of patients and muscles that are going to have a very important impact in your lives, you want to make sure the cells are safe. So the plan here is a small number of patients in the foot, and as soon as we have the green light, we'll be focusing, shifting towards refining of efficacy, motor outcomes, and different muscles. And the goal here is that patients that have been injected in the foot can return and be injected potentially in the hand. So special thanks to my group. It's small but very competent, as I would say. We were able to move this to the clinical grade facility, and here in the lower, a number of collaborators and all their teams. And I would like to thank again the parent project and Duchenne UK for the support. Duchenne UK funded our preclinical studies, and the partnership of PPMD and Duchenne UK now fund the first patients for the phase 1. And thank you all for your attention, and I'll be around for questions if you have any. Thank you.